So first of all, it was a great pleasure for me to have this opportunity of my presentation. Title of the talk is Development of Evaluation Methods of Point to Consider for Industrial Implementation for MPS. And I was very surprised that my interest is, many parts of my interest is overlapping your talk. So, but we have still something new. So let's start my talk. My name is Seiichi Ishida from National Institute of Health Sciences and Seiichi University. I used to have the section chief of the Division of Pharmacology in National Institute of Health Sciences, and currently I continue my research at Seiichi University. I also continue to study in NIHS as a guest researcher. My research interest is to reconstitute hepatic sinusoid in vitro and constructing drug efficacy and safety evaluation system. These are today's agenda. I've been participating in 3 different MPS projects founded by AMEDO, as shown here. AMEDO stands for Japan Agency for Medical Research and Development, and is one of the largest funding agencies in Japan. I'm mainly studying the standardization of MPS from the point of regulatory science. Today, I would like to discuss how we are dealing with the development of evaluation methods of point to consider for industrial implementation of MPS. This is the chart which shows how new test methods research reaches the regulatory acceptance adapted from OECD's document. When the idea of new test method comes to a researcher's mind, he or she begins research and development to turn their idea into a realistic testing method. AMEDO MPS-1 was responsible for this stage and supported by Ministry of Economy, Trade, and Industry. Let me introduce what we have done and what we achieved in the first project. This slide shows the organization of AMEDO MPS-1. This project was launched in 2017 and finished last year. The project consists of 4 programs. Characteristics of this project was the pharmaceutical companies and the device supply companies were also participating for developing models and conducting standardization tests for MPS. And although not shown in the diagram, Pharmaceutics and Medical Device Agency, PMDA, which undertakes the review of pharmaceutics in Japan, participated in the project as an observer and exchanging opinion regularly. As a result of 5 years dedication of participants, 4 MPS are close to the market. Top 1 is the device to evaluate the first pass effect of the absorption in the small intestine and the metabolism in the liver. Second device can supply shear stress by perfusing of culture medium in the upper and lower side of the membrane. Third and fourth devices is to verify the interaction between from 2 to 4 organs by connecting culture chambers. Next, I'd like to introduce what we are discussing for the development of evaluation methods or point to consider for industrial implementation of MPS in AMEDO MPS-2 project. So again, this is the chart which shows how new test methods reaches the regulatory acceptance. AMEDO MPS-1 was responsible for the research and development of the new test method and what is necessary for the next stage is to make MPS have good reproducibility and easy handling. These are important for a variety of users to use MPS in their own needs, that is to say, industrial implementation of MPS. To accomplish the industrial implementation of MPS, AMEDO MPS2 project has started last year. In the case of AMEDO MPS2 project, two model applications are selected. One is the first-pass enterohepatic circulation model using MS plate, and the other is small intestine permeability model employing fluid 3D cross. MS plate, left one, were originally devised by Dr. Matsunaga at Nagoya City University and fit for the evaluation of the first-pass effect of the absorption in the small intestine and the metabolism in the liver. Fluid 3D cross, right one, were originally developed by Dr. Kimura at Tokai University and distributed by Tokyo Oka Kogyo now. It can apply shear stress by perfusing of culture medium in the upper and lower side of membrane. Program 1, led by Dr. Ito at the University of Tsukuba, has selected these two use cases as the model for the implementation of MPS to the industry, based on the hearing of a pharmaceutical company about their unmet needs. Program 3, which I organized, is responsible for the standardization of these MPS applications. Let me explain what we are doing for the development of evaluation method of point to consider for industrial implementation of MPS in the next several slides. This is the process we are thinking necessary for the industrial implementation of the MPS. First part of this process, shown in blue color, is establishment of the context of MPS use case by user. In other words, performance standards of MPS are considered based on the unmet needs that users are expecting for MPS to solve. Second part is the industrial implementation of MPS, shown in red color. Is extraction and verification of consideration points necessary for the regulatory acceptance of MPS. Selection of the model cells and the development of the MPS devices should be according to the performance standards of MPS. To do so, we have to establish the cell criteria and the device standards. Cell criteria and the device standards are consisted of consideration points for each. The example of the consideration point for the cell criteria are like the good functionality, fit for COU on MPS, and so on. And the example of the consideration point for device standards are good supply of oxygen, and so on. After the establishment of the test procedure, there are also some consideration points like reproducibility, etc. Here in the following slides, I'll introduce two consideration points, one for cell and the other for device. This is the result of the paper we published recently. In this paper, we studied the consideration point of commercially available hepatocyte at the cell source for liver MPS by comparing liver characteristics. Left panel shows the distribution of drug metabolism activities in 8 different lots of cryopreserved hepatocytes. As you know, there are several order differences among the 8 lots of hepatocytes. In the right panel, we studied the expression of intestinal markers in human iPS-derived hepatocyte. The expression of intestinal markers was observed in some cases, suggesting that iPS-derived hepatocyte might have some intestinal characteristics. This slide shows the difference of the time for cells to adhere to commercial culture dish and COP, cycloolefin polymers. 2 hours after plating, cells were washed with PVS and number of the cells left on the culture surface was counted. As depicted on the right graph, adherence of cells depends on the materials used. For example, at 2 hours after plating, cell attaches polystyrene plate well, but almost no cells attached to cycloolefin polymers. Thus, it is okay to start medium circulation at 2 hours after plating cells on polystyrene plate, but not good on COP plate. So the timing to start the circulation depends on cell attachment. Example of consideration point for MPS devices is shown in this slide. Upper left photo is the classical Barak type MPS. As shown in the upper right panel, the liquid going into the chamber makes the laminar flow. Thus, the dissolved oxygen in the medium is consumed by the cells on inlet side, and low oxygen medium go down to the outlet side. As a result, the cells growing in the outlet side are exposed to low oxygen medium. This causes more cell death in the outlet side comparing to those in the inlet side. This result shows that the medium flow is important to get the good cell culture condition. I introduced some example of consideration point of the MPS from cell side and device side for the industrial implementation. The more MPS is complicated, the more the number of the consideration point could be. The following is one of the such cases. One of our research goals is continuous recovery of the bile from the bile canonically. There are several reports of co-culture of biliary epithelial cells, sinusoidal epithelial cells, and hepatocytes. But we are thinking that by designing the proper MPS shown left lower panel, will make it possible with less consideration point. There are a variety of scaffold. There are a variety of scaffold available to do the 3D culture of parenchymal cells without co-culture stroma cells. Incorporation of such scaffold in MPS could lead to the simplification of culture condition with less consideration point. Next, let's move to discuss the consideration point from the regulatory side of the MPS development. One of our goals is the regulatory acceptance of MPS. That means that the data obtained from MPS is applicable to the application of the new drug to the authorities. It's not the case in Japan yet. Thus, to get the regulatory acceptance of MPS project has started last year in parallel to the ADME MPS 2. MPS RS stands for the regulatory science, is supported by the Ministry of Health, Labor, and Welfare. To fulfill the process of regulatory acceptance requires the participation of 4 stakeholders: academia, supplier, and supplier company users, mainly pharmaceutical companies and the CRO, and the regulatory sector. In our experience in AMED MPS 1, we found that there are gaps in the understanding and perception of the development and use of MPS among these stakeholders. Thus, the project led by National Institute of Health Sciences was planned, and the MPS RS project was initiated last year. We are summarizing the consideration point as the application note for MPS users in this project now. As shown in the previous slides, the participation of 4 stakeholders: academia, supplier, companies, users, mainly pharmaceutical companies and CROs, and the regulatory sector is important for the discussion of the way to the regulatory acceptance of MPS. Now, we set up the MPS consortium in Japan. The objective of this consortium is promotion of the development of guidelines for test methods using MPS originated in Japan. It just launched, and we are hoping to start a discussion not only in Japan but also with other countries to fulfill this objective. Promotion of the development of guidelines for test method using MPS originated in Japan. Thank you very much for your attention.
Thank you, Dr. Ishida, for this informative presentation. Do we have any questions?
Okay, thank you. So we can move to the next speaker.